Okay, so here we go. Home stretch. This is the last part of one of the nervous system lectures that we'll have. So I've been doing this great big endeavor with you all. And so just to continue off of where we left off, we'll do integration of the autonomic functions, integration and control of autonomic functions. So last time we were here, I finished up the cranial nerves with you all. I talked about the different parts of the brain, and then we talked about the autonomic nervous system kind of as a whole and how it functions for the body. So now we're talking about like integrating the control of it and different autonomic functions. So you'll have autonomic, aka visceral reflexes. And a visceral autonomic reflex adjusts the activity of a visceral effector, often unconsciously. So this just means that the effectors, like the different organs and stuff that the autonomic nervous system will innervate, they will have reflexive actions as a response to a stimulus that you don't have to consciously think about in order to make happen. So some examples of that are like changes in blood pressure, digestive functions, and things of that nature. Also, you think of like the filling and emptying of a bladder or defecation. So autonomic reflexes will occur over autonomic reflex arcs. So remember when we talked about the reflex arcs before, it's kind of similar, but this is specifically for the autonomic nervous system. So some components of that reflex arc are, again, like a sensory receptor, a sensory neuron, an integrating center, pre and post ganglionic motor neurons, and then the visceral effector. So more control about the autonomic nervous system. You're not aware of autonomic responses because the control center is in lower regions of the brain. So these aren't things that you have to consciously think about to make happen. The hypothalamus is one of the major control centers as far as input goes, emotions, and it'll input emotions and visceral sensory information. And some of those things are like smell, taste, temperature, as well as like osmolarity of the blood, things like that. And what it will output, it sends things to the nuclei in the brainstem and the spinal cord. So there are posterior and lateral portions of the hypothalamus, though, and those control the sympathetic aspect of the nervous system. So remember, your sympathetic system is your fight or flight. So that's going to be like increased heart rate, inhibition of the GI tract, because it's not like organs that need stimulation during an emergency, and increased temperature. The anterior and medial portions of the hypothalamus are going to control the parasympathetic nervous system. And so that's your decreased heart rate, your lowered blood pressure, increased GI tract secretion, and mobility. So autonomic dysreflexia. So what is autonomic dysreflexia? That's an exaggerated response to the sympathetic nervous system in cases of like the spinal cord injury above T6. So remember that sympathetic response is going to happen in your thoracolumbar region, but if there's an injury to the spinal cord that's happening above T6, you can have an exaggerated sympathetic response. Certain sensory impulses will trigger mass stimulation of sympathetic nerves that happen below the injury. And so as a result, you'll get vasoconstriction, and that's going to elevate your blood pressure, because think if your blood vessels are smaller, but you're still trying to pump the same amount of blood through them, the pressure on that tube is going to be higher. So vasoconstriction leads to elevated blood pressure. The parasympathetic nervous system will attempt to compensate for the sympathetic nervous system going into overdrive, and so it will try to slow the heart rate down and dilate the blood vessels above the injury. And so sometimes that can result in like pounding headaches, sweating, and warm skin above the injury and cool, dry skin below. So you're having, honestly, like these dual responses where you shouldn't normally be having them. So because of this, you can have seizures, strokes, and even heart attacks. And now we'll get into rabies. So this is one of the big neurological disorders that is discussed in animals. So this is a really important one that we'll get a lot into. So just some general information about rabies. Rabies is a viral infection of the central nervous system, and it causes encephalitis. So using, your medical terminology, breaking down that word, itis means inflammation, in is inside, and ceph is often noted for heads. So that means that there's an inflammation of like the inside of the head. Rabies is normally contracted from infected animal bites. So an animal that has rabies, it bites another animal and it can give it to the other one. This is nearly always fatal without PEP. So this is that prophylactic drug that they give in order to prevent rabies from taking post-exposure prophylaxis. And so treatment of the bite, wound, and rabies vaccine shots are what's necessary in order to prevent an infection from taking place. PEP should be given to all people with a scratch, bite, or mucous membrane exposure to infected animals unless the animal tests negative for rabies. So that's why veterinary professionals, pretty much anybody that works with animals, has to get a rabies shot, because every once in a while, you'll encounter some animals that you don't know exactly where they're from. And so if they bite you and break the skin and you have like mucous membranes exposed, you could potentially become infected with rabies. For example, like if you handle a down bat, like a bat that's down, especially because bats are big vectors for rabies, PEP should be given. So what's the pathology of rabies? So you're specifically going to look at the Negri bodies in rabies virus, right? So this is an example of one of them. This is a Negri body. And so immunohistochemistry is the test that's done in order to target the viral antigen that's in rabies in order for you to be able to diagnose it. So you can use immunohistochemistry and able to diagnose rabies being present in animals. This is just a picture in order to be able to describe to you that process that we talked about. So an infected animal can bite a person or another animal. If it breaks the skin and exposes that saliva to the mucous membranes, since the virus is transmitted by infected saliva, it can travel all the way to the brain and cause that brain inflammation, and that's that encephalitis that we discussed. That is a main hallmark of a rabies infection. So more just general rabies information. The incubation period for this disease is usually several weeks, but it can go out to months. The virus will pass via the peripheral nervous system. So that's via the sensory and motor nerve axons that are at the bite area and then travel up through the CNS. But once it's in the CNS, the virus replicates. And so the animal becomes infectious after the virus has replicated in the brain and disseminated. But that virus will disseminate to the salivary glands and other organs via the nerve. So just some more general information for you. Here are some images. The rabies virus is an RNA virus that has a distinct bullet shape. So this is that bullet shape that you'll refer to. Looking at rabies, you can see like actual pictures of it. That's what it looks like. And so there's this cycle of infection and replication that takes place. So the fusion of the rabies virus envelope to the host cell membrane is what initiates the infection process. So like the rabies virus is going to match on to the cells of the animal that it's trying to infect, right? And the virus will penetrate the host cell and enter the cytoplasm by pinocytosis. So that's essentially it being like taken in with like liquid particles, right? And so this will happen where the membrane surrounds the invading rabies virus and it will bring it into the cell itself. And so then the rabies virus essentially will like uncoat, so that way the envelope that's surrounding the virus will disappear, so that way the rabies virus RNA that's within its cell can be released into the cytoplasm of the host cell, right? And so the messenger RNA will be transcribed in order to permit virus replication. So then once that translation takes place, so it'll make like the rabies proteins, and that happens in the ribosomes of the cytoplasm. I didn't say that. But once that happens, the replication of the viral genome can start. And so that first step in viral replication is the synthesis of full-length copies of the viral genome. So when it switches, when it starts replicating, essentially the RNA transcription becomes non-stop. So normally, like you have a cell and like it's transcribing things and replicating, and so there are stop and start codons in order for it to know when to, start and stop this process. But when it starts transcribing the rabies virus, those stop codons are ignored, so it will just keep replicating them. And so then you'll have these strands of rabies RNA that's present. And so this serves as a template for more strands of the virus in order to be replicated. So that complete virus then, once it's been replicated, comes out of the cell membrane, and it's present within the central nervous system now. So it can start to infect other areas of it. Viral budding will take place, and that viral budding will go into the salivary glands. And so also, with the virus being present in the CNS, it starts to create this aggressive behavior, right? And so the animals will start to bite things. And so now that it's present within the salivary glands, if it bites another animal and that saliva gets exposed to that host animal's mucosa, it maximizes the chance of another viral infection in a new host. And so it's really kind of insidious, being able to manipulate the animals into having more aggressive behavior and to better spread the virus along. So here's the infection path in a raccoon, right? It's kind of what we discussed before. The animal is going to be bitten by a rabid animal. The rabies virus will enter through the infected saliva. That virus will spread through the nerves to the brain and the spinal cord, where the virus will incubate for like 3 to 12 weeks, which is really kind of a long time. And then the virus in the brain will enter the brain cells and multiply, like we discussed on the last slide. And then the virus will pass to the salivary glands, and then the animal will begin to show symptoms. And then usually the animal will die within 7 days after becoming symptomatic. And so that's it in like raccoons, and basically like a solid template for how it is with most animals. But signs and symptoms in humans sometimes look a little bit different. So you'll have like flu-like symptoms, so that malaise, which is that general feeling of kind of like unwellness, like when you're just like, like that's what malaise is, as well as fever or headache may occur. The bite will heal, and then later on the area just might be kind of uncomfortable. But then you'll start to see cerebral dysfunction occur. So you can tell that it's cerebral dysfunction because it'll be noted by like anxiety, confusion, agitation, delirium, abnormal behavior, hallucinations, hypersalivation, jaw and pharynx muscle contractions, muscle spasms, paralysis, and insomnia. This happens about 25 days usually after a person gets bitten and becomes infected. There's this acute period of disease that ends like 2 to 10 days after all of this happens, and it usually ends with a coma and death. So like if all of these signs start to show up, it's looking bad. Shortly afterwards, a coma and death will follow. So there have been 6 recorded cases. Normally, like we said, rabies is fatal. Most of the cases that are reported with it wind up leading to the human or animal that's infected dying. But there are a couple cases where people have lived. But to date, there have only been 6 cases where human survival from clinical rabies has occurred. So it's clinical rabies, meaning like they've begun to show and develop symptoms. And so in each one, the people have been exposed to either pre or post-exposure prophylaxis. So basically, this is kind of saying that like your chances of survival are not very high, but if you are going to survive, it's definitely going to have something to do with pre and post-exposure prophylaxis. Nobody has survived without it. Well, except for now. I'm just saying that like there has been one case of survival with none involved. But rabies is very much so considered a deadly virus. I would not risk the chances of it if you can get away from it. As far as pathology goes, like we discussed, there's an encephalitis and a myelitis that will occur. So this is inflammation of the brain, and myelitis is like inflammation of like that myelin. So there's perivascular infiltration with lymphocytes and polymorphonuclear leukocytes and the plasma cells throughout the entire CNS. So remember, we talked about lymphocytes. Those are cells for like the lymphatic system, as well as the polymorphonuclear leukocytes, so your white blood cells, and plasma cells throughout the entire CNS. So basically, it sees an infection trying to occur, and it's attempting to fight it off. Rabies often causes cytoplasmic eosinophilic inclusion bodies called Negri bodies. That's what we discussed before, and they're in the cells of the cerebellum specifically. These inclusions have been identified as areas of active viral replication. So an eosinophilic inclusion body, that's, we talked about different stains before, like the eosin stain. So it's going to show up that like pink color an inclusion body within the cytoplasm of an infected cell. And so they're going to call that a Negri body. And those cells are going to be present in the cerebellum. And these inclusions are what people generally consider as an area of active replication of the rabies virus. So again, that's where the cerebellum is in people. So that's where you would find that tissue if you were looking, trying to do immunohistochemistry to determine if rabies was present. So this is just giving further depiction of like where it might be. This is a Negri body. So again, like we discussed, eosinophilic, like it's pink, and this is your entire cell. So this like eosinophilic inclusion body in the cytoplasm. Remember, intracytoplasmic inclusion bodies are ones that are going to be within the cytoplasm of the cell. So what's the outcome of rabies exposure? The virus variant has a determination of it. The dose of the virus that is present, like through the bite wounds, the route and location of exposure, as well as the individual host factors like age and immune defenses will all determine what the outcome of a disease infection might be. So, it like depends on what animal it came from, how bad the bite was, how close to the brain the bite was, are all things that can determine like how well the body could fight it off before it gets to the CNS and has major effects. So this is a way to determine how cerebrospinal fluid can be collected. So CSF, cerebrospinal fluid, is often a test sample that can help to measure the pressure in the fluid that surrounds the brain, right? So cerebrospinal fluid normally acts like a cushion that protects the brain from injury. And so the fluid is normally clear, right? In order to get it, the patient has to lay on their side with like knees pulled up towards the chest and your chin tucked downward like that. And so sometimes they kind of test it in different ways, but after that, they clean it up, and so they'll get this like a numbing agent in order to go in, and they'll insert the needle usually between the 3rd and 4th lumbar vertebrae in people. And once that needle is properly positioned, the spinal fluid is measured and collected, and then you remove the needle and clean it up and put a bandage over it, and usually wait after while the test is being done, and they will evaluate like the pressure sensation from when the needle is inserted, and then the pain should normally go away, but the pressure should normally be between 50 to 180 like millimeters of mercury or whatever. And so like the normal cerebrospinal fluid should be like this clear, colorless kind of consistency, a little thick. But when something is affected, you'll have variations in it. Like it could be a different color or different consistency or pressure that would affect it. But what other lab tests are done in regards to rabies? So DFA is the standard test. Infected animal tissues will have like the rabies virus antigens present. And so the ideal tissue to test is the brain, which is why this is such a serious disease, because like you cannot get brain tissue without killing the host, right? So the DFA test that's done contains a fluorescently labeled anti-rabies antibody. And so the brain tissue is incubated with the kit antibody that it comes from, right? And so that rabies antigen and antibody will bind if rabies is present. And then, it washes it out. And so a fluorescent microscope is used to display the results. So if it's positive, you'll have fluorescent markers like this, where it's kind of like these glow-in-the-dark sort of shapes that pop up for positive fluorescence. If it's negative, it'll just look dark. So what are some amplification methods in order to be able to determine if the virus is there? So like virus particles are very, very small, and sometimes are hard to determine in different tissues. So you want to be able to amplify them if you're testing for rabies specifically. So virus isolation in cell cultures increases the virus concentration. Like if you isolate the virus, you know that there aren't going to be other things there. So it's concentrated in whatever specific area you've isolated it in, right? Mouse neuroblastoma cells and baby hamster kidney cells are often used for viral culture. So these are positive bands on PCR where you can see that it's present. So using PCR, the rabies virus nucleic acid is enzymatically amplified as DNA copies. That rabies RNA is then copied into DNA molecules using reverse transcriptase. And so then the DNA copies of the rabies can then be amplified using PCR, or polymerase chain reaction. And so people will use this in order to confirm DFA results, and that can be used on like saliva and skin biopsy samples. This is an example of like a human rabies patient. You can see that they have him chained down to the bed because he's probably exhibiting a lot of erratic and like aggressive behaviors, and they don't want him to get up and potentially infect anyone else. Remember, other animals besides dogs and cats can get rabies too. In fact, in North America, it's most commonly seen in skunks, foxes, bats, raccoons, and other warm-blooded animals. But like cows and ferrets can also get it, as well as small rodents, lagomorphs like rabbits and hares. But small rodents and these lagomorphs are often not found with it. It's hard to find them with it, and have not been known to cause rabies infections in people. Those other animals have also passed on like human transmission. At least not in the US. They haven't been seen with that. Bites by these animals are usually not considered a risk of rabies unless the animal was already sick or behaving in like some unusual kind of way, or if like rabies is widespread within like that general area. So if it's endemic to that particular population. So how would you control rabies? So like they control wildlife rabies through like bite technology, in order to, and also, control populations. That's why like, you vaccinate your pets and things like that in order to prevent from like local exposures, but they can like analyze like bites and stuff that were taking place to see if rabies is in the area and things like that. So this is a woodchuck. I know they were talking about like there was like a breakout in like 1985, 84 of like woodchucks with rabies in a particular area. Here is a bat. It's very small, but that's all it takes sometimes. Bats can be really small or really large, but bats are one of the main known hosts for rabies, so you have to be careful whenever you're like handling or around exposure to them. Side note, I know that there are like lots of bats around here in Tuskegee, so every once in a while some like pop up in buildings or whatever. So if you ever see one, especially one that's just like dead or something, call somebody to come get it, because there's no need to unnecessarily expose yourself to rabies or any other potential diseases that like animals could have. So you can notice the size of the teeth, like in this particular case, this is a really small bat, so it's got really small teeth. So you might not even notice a bite from this animal if you didn't see it, right? So bites can be microscopic, and so you might not even notice that they're present. But if saliva comes in contact with it, that bite can still be infected. However, on the flip side, you've got this Mexican free-tail bat, and it looks enormous, you know? So what you do in case of a bite, you should wash the wound out immediately with soap and water and treat with betadine. Betadine is like a sterile scrub. Depending on, the animal that bit you, and if it's possible, try to capture it and quarantine it in order to watch for like signs or symptoms that may pop up. Contact local or state public health officials in order to have the animal tested. Like if it's like a fox or something and you trap it and you don't necessarily care about keeping it alive, whether it's got rabies or not, calling somebody to go ahead and come in and test it, and so that way you can be in the clear potentially sooner, by finding out that information quicker. Visit a human doctor, whatever doctor is fitting for the thing that got bitten, whether it be a person or an animal. Go to that appropriate physician in order to get analyzed, and then treat the wound with rabies immune globulin. So that's that RIG. And so this is like a preparation of antibodies that's made from plasma or serum from selected human donors who have been immunized with the rabies vaccine before. And so this is injected around the wound. And so the wound, or, people might irrigate the wound specifically with it. Some other stuff that happens if you get bitten, the patient is also given a shot of the rabies vaccine. So that's kind of like, that's that post-exposure prophylaxis that you're exposed to. 4 or more rabies vaccines are given at day 3, day 7, day 14, and day 28. So like if you get bitten, they will give more vaccines at that interval to help out. Just for your information, the rabies vaccine was made by Louis Pasteur back in 1884. And so it was made from like desiccated brain and spinal cords from infected rabbits. And then that tissue was treated with formalin, ether, and that killed the virus. And so this used to be a vaccine that like would be injected once a day for like 25 days in your stomach. So that's like a much more aggressive treatment. But like now, they'll give you the vaccine in your arm and it takes like 4 times or whatever. So that's why you've got like the 4 shots given like at those different day intervals. But all rabies vaccines are given in that deltoid muscle. You guys know where that is now. So these are like reported cases of rabies by bats, rabies from bats, like by county. So you can see there are areas where it's a lot more popular, especially like in the red specifically, but that's where like rabid bats were, but like it's been tested in all these different areas. So you might not see it in some of these like places, but also the population density is a lot smaller there. So people might be able to avoid certain situations, but like there is it's nationwide. You can see that like there's some areas where you'll see it a little bit more, but it is distributed all throughout the country. So cases have gone up in recent years from like wild animals, but down from domestic animals. And so a lot of that comes from the fact that like we vaccinate domestic animals now. That's like a core vaccine when you have pets. And so that really does help the spread from that level. But as there are more people on the planet and people going out and doing wild activities or whatever, they're exposed to more wild animals. So like cases of rabies from wild animals have gone up. So what are some FDA and CDC requirements? They don't test for rabies on like transplants that are going into bodies. It's not an effective test taking tool, right? Well, it's not an effective tool because it takes so long, for symptoms or things to show up or for like that replication process. So they kind of avoid it. So really the best measures that you can do is to make sure that you have, if you're working in this animal industry or whatever, to have appropriate vaccinations. These are references for all of the nervous system stuff that we've discussed, but this is the end of the nervous system. We end on a high note, sort of, if you think about it, with rabies. But that's all that I have for you all as far as these topics go. Please, 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 if you have any questions at all, ask about them. Email me or Jade or post things to the discussion board, because I know the nervous system is dense and it is a lot of information, but you're still responsible for it. So, make sure you read the book if you have any questions. Come to us if you have any questions, and we will try to get that sorted for you all. So with that, good luck.